The really interesting things about this new paper that come to light are that we provide two new opportunities. The first is that it provides a computational method to deal with deep binding pockets. So this is treating binding pockets not just as one single unit, but looking at the subtlety of how molecules interact as they pass down into a deep protein pocket. The second and important feature is that we've used the small plant hormone auxin as a case study, and we've used the method to illuminate how different parts of that deep binding pocket contribute to selectivity for this hormone.
Docking is a useful computational tool that generates poses and ligands of small molecules in their receptor site. We apply this methodology to the study of the plant hormone auxin in its receptor tier 1. Tier 1 does not have a shallow binding pocket, but rather a deep binding pocket, and what we found is that we had to include or factor in this deep binding pocket to account for the passage of ligands towards the bottom of this binding site, and as such we developed the concept of TomoDock.
The idea about TomoDock came from the realization that we are dealing with two different length scales in this problem. One was the length scale of the small molecule, which was several angstroms. The other was the length scale of the protein pocket, which was several tens of angstroms. It was logical to expect that the small molecule is not going to materialize at the bottom of the pocket, but rather will take this distance in several steps. In a loose analogy, a person that is trying to get to the bottom of a well safely is not going to plunge themselves down, but are going to try to use their own dexterity and the walls of the well in order to safely descend in multiple steps. The moving search space of TomoDock is supposed to represent exactly this concept, trying to see what happens with the molecule descent on the sequence of steps.
From this work, the main take-home message is that the interaction of auxin and its receptor tier 1 does not happen in a single step. Rather, what we find is that it is a three-step mechanism. First, the first step is the interaction of auxin with a structural feature that is found at approximately the beginning of the deep binding pocket of tier 1, and this is what we call the engagement niche. After auxin has interacted with the engagement niche, orienting its carboxylic tail into it, it proceeds via a sliding down motion of its indole ring that passes between some residues, forming what we call a molecular filter. This molecular filter is a second structural feature of the pocket, and we call it a filter because its action is to allow the biologically active molecules to pass through while inhibiting biologically inactive molecules, for instance tryptophan, from proceeding down towards the bottom of the pocket. Finally, the third step is the actual binding of auxin at the bottom of the pocket onto the receptor tier 1.
The only two requirements if you want to use TomoDock on a system is that you have a protein with a deep structural feature into which functionally a small organic molecule binds. There are very many proteins like this in nature, both in plant and in the animal kingdom, and also human proteins. The very interesting class of such proteins are the transmembrane transport proteins. There, they have to move a small organic molecule from one side of the membrane to the other, and by virtue of its function, they have very, very deep structural features. To this class of protein also belong very important proteins, such as the proteins responsible for the multi-drug resistance in some cancers, or the proteins responsible for antibiotic resistance in bacteria. As far as we are concerned, we are very interested to apply TomoDock to the transport proteins for auxin and have a complete picture how auxin transport and perception in the plant cell works.